Hi Year 12s, welcome to our next video looking at chapter 1.8, which is biotechnology applications. So our SACE key ideas for this subchapter is we need to know that biotechnology can involve the use of plasmids and viruses as vectors, bacterial enzymes and yeasts. Techniques can include bacterial transformations, electroporation and microinjection. So this really relates to the previous chapter as well, chapter 1.7. So you need to be able to describe how particular genes can be selected using probes and removed using restriction enzymes. Describe how selected genes can be transferred between species. Describe how CRISPR can be used to edit and/or transfer genes, and discuss the design of new proteins and their uses. So to start off with, we're going to be looking at transgenic organisms, cloning and therapeutic cloning, and then we're going to look at some social and ethical issues before looking at CRISPR. Okay, so firstly, just some clarification between the difference between transgenic and genetically modified organisms. So transgenic organisms have had foreign DNA inserted into their genome so that they can express different characteristics and produce different proteins. Genetically modified organisms have had their own genes either switched on or off. So no foreign DNA has been inserted into a GMO. So all transgenic organisms are GMOs because they have been genetically modified, but not all GMOs are transgenic because they have not necessarily had foreign DNA inserted into them. Okay, so there are 4 steps to creating a transgenic organism. Firstly, we need to identify a gene of interest. For example, that might be one that confers herbicide resistance in a plant. Step 2 is isolate this gene using a probe and a specific restriction enzyme. So remember, the probe has nucleotides that are complementary to the gene of interest, and restriction enzyme will cut that gene. We insert the gene into the new genome, and then we grow or develop that transgenic organism so that it will replicate that DNA, that transgenic DNA, and express that gene by synthesizing proteins. So here is a diagram, and you can find this on page 107 of your workbook. So I'm just going to go through those 4 steps again. So in the cell containing the gene of interest, we will need to extract the DNA, locate that gene of interest, and then isolate that gene using probes and a restriction enzyme. We need to do the same with a plasmid, for example. So we would need to isolate that plasmid from bacteria and then use the same restriction enzyme to cut the plasmid in the same place. We insert the gene into the plasmid using a range of gene transfer techniques that were discussed in chapter 1.7. We insert that plasmid into a bacterial cell that contains the recombinant bacterium. So this is the transgenic organism. The cells will replicate with that gene of interest, and then we can either isolate that protein, for example, for insulin for diabetics, or we might actually copy the gene, because now we've got heaps of copies of that gene, and use it. So for example, we might insert it into rice so that it contains the gene to produce vitamin A, or altering bacteria so that the bacteria can clear up oil spills in the ocean. So here are 2 examples of transgenic organisms. The first one is transgenic tomatoes, and these were commercially known as flavor saver tomatoes. So these tomatoes had a gene inserted that changed the expression of the gene responsible for softening of the cell wall. So that means the cell wall was harder. That meant that tomatoes could be shipped over greater distances and stored for longer periods without going soft and going off. Another example is golden rice. This is a variety of rice which has incorporated the gene for vitamin A production. So electroporation and agrobacterium, microparticle gun and viral vector are all used to create golden rice. Vitamin A deficiency is a leading cause of childhood blindness, and because rice is a staple food crop for over half of the world's population, and making up 30 to 72% of the energy intake for people in Asian countries, this is the perfect crop for targeting vitamin deficiency. So that's why this gene to produce vitamin A in rice was developed, and that means that there's less vitamin A deficiency, and that means that there would be less childhood blindness. Here is some more examples of transgenic plant features and their benefits. So we could create transgenic organisms that have improved uptake of minerals by the plant, and this reduces the need for fertilizers. We could improve the ability to withstand drought, and this means that crops can be grown in unfavorable conditions. We can improve the resistance to disease, reducing the need for pesticide use. We can control the ripening of fruit, which means there will be less waste and reduced loss of fruit after harvesting. And improved frost resistance. So growing and harvest season can be increased. So during the colder weather, the plants are resistant to that. Okay, the next biotechnology application is cloning. So a clone is a genetically identical organism or a group of genetically identical cells derived from a single parent cell, produced asexually. So you might have heard of Dolly, who is one of the most famous examples of a cloned organism. Dolly the sheep was cloned late in the 1990s using the nucleus in a cell from the udder of a fin dorsate ewe. And so the nucleus from the fin dorsate ewe contained the genetic information that was then placed in a Scottish blackface ewe's egg cell. And then this surrogate mother gave birth to Dolly, and that Dolly was a clone of the original fawn Dorset sheep. So the steps for cloning are very similar to what we've been through with transgenic organisms, but the first step is to isolate DNA from a somatic cell of a donor individual. So isolating DNA from a cell other than a sperm or an egg. Then we transfer that DNA into an unfertilized egg cell that has had the genetic material removed. That cell is given an electric pulse to stimulate division, and then that embryo is implanted into a surrogate mother, and the surrogate mother gives birth to an exact copy of that donor individual. So here's a diagram looking at cloning of an animal. So if we have a donor cell taken, so that, remember, that's a somatic cell, and then we have an egg cell, and the two cells are fused so that this egg cell becomes fertilized, and then that fused cell will divide to form an embryo. The embryo is placed in the uterus of a foster or a surrogate mother, and then the embryo develops normally and is born and is an exact copy or clone of that donor animal that we started with. Therapeutic cloning is the use of cloned stem cells in medicine. So stem cells can be obtained by either a fertilized ovum, so from IVF, from bone marrow or the umbilical cord blood, or we can reprogram somatic cells to become stem cells. So stem cells can be used to produce specific tissues in the lab and then transplanted into patients to replace or repair damaged tissue in the body. Injecting stem cells does carry risks such as rejection, development of wrong tissue, or tumours. Okay, we're now going to look at social and ethical issues of these applications. So the first one is bioethics. So this is the study of biological ethical issues. Genetic engineering offers unlimited potential, from improving food availability and nutritional value to disease prevention and cure, but there's a question of should we be doing this? So related to agriculture, animals may suffer when expressing the new gene, there may be an allergic reaction, transgenic products may have toxic effects that are as yet unknown, and transgenic crops may be so successful they overtake the native vegetation, and this reduces biodiversity. Related to medicine, there could be unknown side effects for individuals. Large companies who hold the patents may control the knowledge and products, and so that would make those that technology very expensive and those products very expensive. Individuals may manipulate their children to have more desirable characteristics, and insurance companies may access the genome of an individual and deny medical coverage. The ethics of gene manipulation. So DNA manipulation has raised serious concern about the impacts of it. For example, genetically modified foods and transgenic crops may have unforeseen effects on the ecosystem. So here are some arguments for gene manipulation and arguments against. The pros, or the arguments for gene manipulation, are the known short-term benefits that come from manipulating genes. Better quality harvests, less need for fertilizers, leads to an economic profit, may help in treating genetic diseases and assisting with paternity and criminal cases. Arguments against. So there's unknown long-term costs from manipulating DNA. It might lead to loss of biodiversity, potentially harmful effects on the ecosystem. It's unnatural. There might be environmental and social costs. Certain biotechnologies are expensive, so that's economic loss. And genetic information might be used against individuals. Collecting and storing genetic information also has some advantages and disadvantages. So advantages are that geneticists can increase their understanding of genetics and the genome, human genome, assist in curing genetic diseases, targeted health cures for individuals. If you know your genetics, then your health practice can be tailored to your genetics. It can help with tracking missing persons and preventing crime, and genetic epidemiology regarding genes and the environment. So that's to do with disease. Disadvantages: discrimination, for example, being susceptible to disease, economic costs of collection and storage, making errors and maybe convicting innocent people. Who has access to this information? Is it stored in a computer cloud? Is it possible that it could be hacked into? There might be race discrimination if your genetics reveal different nationalities, and refusal to insure people who have a certain susceptibility to disease. So lastly, gene therapy. So this involves changing the body's set of genetic instructions and raises many unique ethical concerns. So who decides what is normal and what is a disability or a disorder? Will the high costs of gene therapy make it available only to wealthy people, and then that would impact our social class? Could the widespread use of gene therapy make society less accepting of people who are different? And should people be allowed to use gene therapy to enhance basic human traits such as high intelligence or athletic ability? So gene therapy raises quite a lot of ethical concerns. Some arguments for gene therapy are that patients wouldn't be required to continually get a supply of medicine. It's a once-only treatment, so it's not going to interfere with their lifestyle. There would be less absenteeism for work. So if you're sick less often, you can go into work, which means companies have higher productivity, lower public health costs, money can be diverted into other areas, and fewer sick people would improve the economy. Arguments against gene therapy is that people may be refused insurance due to diagnosis of genetic disease. We've touched on that a few times. People may be discriminated against their employers due to genetic diseases, so discriminated by their employers. Lack of access to technology for underdeveloped countries might increase that divide between developed countries and underdeveloped countries. There's religious and cultural opposition to gene therapy, and it could have effects on evolution, blurring boundaries between species. These are some examples where the use of technology has proven to be highly beneficial, and in particular manufacturing proteins. So vaccines have been very beneficial to our society all around the world. Also developing hormones such as insulin and HCG. There are many therapeutic drugs, biofuels, which are better for the environment, food supplements, so a lot of vitamins, and minerals and restriction enzymes. So that leads us into talking about CRISPR, which uses the Cas9 restriction enzyme. So CRISPR is a new gene editing technique that can be applied to DNA of any species, and there are clear benefits to healthcare and agriculture. One example of its use is that it's already been used to genetically engineer mosquitoes to reduce the spread of malaria. So the Cas9 enzyme is produced to bind and cleave the DNA without making mistakes and cutting the DNA at other sites. So it's very reliable. And there is an RNA guide which guides the Cas9 enzyme to cut the DNA in the right position. So these are two diagrams that help us understand how CRISPR works. So if we have this Cas9 enzyme which cuts DNA and the guide RNA which directs the Cas9 to the target DNA, they stick together and then they look for the target DNA. The Cas9 will unzip the target DNA, the guide RNA will match up because it's got a complementary sequence, and if the match is complete, then the Cas9 will cut the target DNA. And then once it's cut, the DNA can be disabled or altered, or it can be edited as well. Here is another diagram looking at how CRISPR works naturally in bacteria. So when a virus invades a bacterial cell, a new spacer is derived from the virus and integrated into the CRISPR sequence, which is already in the bacteria. The CRISPR RNA is formed, and then that guides molecular machinery to target and destroy the viral genome. So CRISPR occurs naturally in bacteria to attack viruses. That's the end of this video, but I am going to link another YouTube video which goes into a bit more detail on CRISPR that I really recommend you also watch and take notes from.